One of the most challenging experiences in medicine is to tell a patient that they have a glioblastoma. Despite recent advances in systemic and immunotherapies, this diagnosis still has a very high rate of morbidity and mortality. But what if I could tell you that we can use the body's own delivery system to transport therapeutics straight to the tumor cells? My name is Emily, I'm an MS3, and I work in the homuncular lab, and I will be presenting today on exosomes and glioma, vehicles for immune modulation and nanotherapeutics. In this presentation, I will talk about the current research on exosomes and their interactions with the immune system. I will be sharing some exciting work coming out of the homuncular lab about this topic. Additionally, I will give you a sneak preview of clinical applications of exosomes, including in biomarkers and in therapeutic applications. So, what are exosomes? Exosomes are extracellular lipid-enclosed vehicles that are secreted by almost all types of cells, including neurons and tumor cells. They are between 40 and 160 nanometers in diameter, and they're formed by the endosomal trafficking system. During their production process, the cell packages various forms of cargo both within and around the exosomes. This cargo includes proteins, RNA, DNA, amino acids, and metabolites. These exosomes then deliver this cargo to target cells and change those target cell behavior. Thus, exosomes function as a form of intercellular communication. Cancer cells also release a lot of exosomes. These exosomes carry many different types of proteins and DNA, and these exosomes are used by the cancer cells to communicate with both the adaptive and innate immune systems. Much work has already been done in this area. For example, PD-L1 and melanoma cell-derived exosomes were shown to suppress CD8 T cells. Breast cancer-derived exosomes carry DNA, which activate dendritic cells. So here we see that exosomes can have many different types of effects, depending on their cargo and their target cell. Ovarian cancer exosomes carry microRNA that promote a tumor-associated macrophage phenotype. With all of this, we wondered, what is the role of exosomes in glioma? And that is the topic of research of the homuncular lab. We in particular are interested in IDH-mutated gliomas because they display an impressive immunosuppressive phenotype. IDH-mutated gliomas are known to release an oncometabolite called 2-HG. 2-HG has been shown to decrease CD8 T cell infiltration and promote an immunosuppressive macrophage phenotype. However, the immunosuppression seen in IDH-mutated gliomas is far more wide-reaching than this. When we analyzed immune cell populations in wild-type and mutant IDH tumors, we see a dramatic difference in the amount of immune cells in these types of tumors, as you can see in this graph here, where the wild-type IDH tumors are shown in blue, and the red represents immune cell populations derived from mutant IDH populations. So we hypothesized that exosomes might be responsible for this effect we're seeing. To investigate this, we isolated tumor exosomes from both wild-type IDH and mutant IDH tumors. We then conducted next-generation sequencing on the contents of those exosomes and looked at differential expression of immune-related genes. What we found were that genes related to immune activation, for example, ULBP1, ULBP3, RBP1, and various chemokines, were all downregulated in the mutant IDH exosomes. We then compared this expression profile between tumor exosomes and their parent-releasing cells, and we found that the profiles were incredibly similar. So essentially, these tumors are secreting tiny little copies of themselves out into the environment. The next question was, what effect do these exosomes have at all? So to do that, we turned to a glioma mouse model. We first isolated IDH mutant exosomes and injected them into mice with IDH wild-type tumors. And the question was, Does the immune environment look more like IDH wild type or IDH mutant? And the answer is that they looked more like IDH mutant. So it matched the phenotype of the exosomes rather than the implanted tumor. We also did the experiment in reverse, so injecting wild type IDH tumor exosomes into mice that had a mutant IDH tumor, and we saw the same thing. The immune environment became molded by the exosomes. We also looked at immune populations both locally, so tumor infiltrating lymphocytes, and also immune populations in the peripheral circulating system. And we saw a similar effect in both. This is incredible because it suggests that exosomes can radically transform the immune system, both on a local and systemic level. To further support this, we looked at the biodistribution of the exosomes. First, we can see the exosomes have a really good penetration into the brain, which will become important when I talk about clinical applications. Furthermore, we can see that the exosomes go all over the body. And so this supports the idea that exosomes are acting globally inside the body. With all this research, our focus is really on clinical applications. And one of those applications is in a non-invasive way to diagnose glioma. Imagine being able to take blood or CSF from a patient and then be able to tell them that they have a glioma and be able to track their progression or recurrence, rather than sticking a needle into their brain and getting a brain biopsy. Several groups are already working on this. One group found that exosome levels of microRNA-21 from CSF collected by lumbar puncture was associated with a glioma diagnosis. Furthermore, the levels of this microRNA were correlated with the grade of the glioma as well. So, using this technology in the future, we could diagnose a patient and also give them prognostic information as well. We can also use exosomes for treatment. Exosomes are ideal drug delivery carriers because they have several unique characteristics. One, they have innate homing properties. Two, they are less toxic than other manufactured drug delivery systems, such as liposomes, which are already in clinical use. And three, they have minimal immune clearance, so they stay in the body for longer. One group has shown proof of concept by creating these neutrophil-derived exosomes. They then loaded these exosomes up with doxorubicin, which is the chemotherapeutic, and then injected them into a mice tumor model. They were able to show that these exosomes were able to efficiently penetrate the blood-brain barrier, and then these exosomes preferentially targeted the tumor while leaving healthy tissue alone. This group showed that the tumor was able to be reduced and there was survival benefit. This is really the future of exosomes. In conclusion, I want to leave you with two main ideas. One, exosomes play a key role in modulating the local and systemic immune environment. And two, exosomes have special properties that we can harness in order to eradicate cancer. I want to thank Dr. Stephen Bram and Dr. Nduka Amankalor, as well as the rest of the Penn Medicine Neurosurgery Department for their assistance in this talk. Thank you so much.